The SaskOilseeds disease testing program is a free program. It's available to all canola growers in the province. We can test for blackleg race ID, so find out what kind of blackleg is it. If you have blackleg in your field, what kind of blackleg, as well as Verticillium Stripe. And then we also offer a soil test for Clubroot to find out if Clubroot is in your soil.
The first thing we do when we receive a stem sample for the SaskOilseeds testing program is we will take our cross section from the bottom of the stem and plate it on a special media that encourages the growth of blackleg. The stem is cut kind of at the base, right between the hypochondral and crown. It's where the highest level of the pathogen is present. We surface sterilize it for a couple of minutes in a mild bleach solution just to get any of the sapharites or any bacterial colonies that are probably on there off of there. And we place it on a special media. It's called V8 auger. It encourages the growth of blackleg and kind of discourages the growth of any bacterial cultures or some of the other things that'll come out of the stems. Those cultures are left for a week to 10 days to grow. Blackleg's fairly slow growing. And at the end of 10 days, all of the cultures that grow on the plate are visually assessed. And anything that looks like it might be blackleg, either the virulent form Labratories maculans or the avirulent form Labratories blackleg, a portion of the culture is removed and a DNA extraction is done directly from the culture. Once we have that DNA, we run a Labratories maculans or Labratories blackleg specific molecular marker against it, and that will tell us what each culture is. All of the cultures that are Labratories maculans then go through another PCR process where we run the 8 race ID markers on them. Those markers give us the avirulent genes that are present in each blackleg. And when we put them together, it gives you the code that kind of explains what's happening in that culture. And you'll use that data to make your seed selection for future years, matching the seeds that have resistance to the specific races of blackleg that could be on your farm. After the stems are cut for the blackleg, this is what they look like after about a week on the auger. So at this point, we'll go through, and anything that resembles blackleg, which would be these 3 cultures here, we'll take scrapings of the pycnidia, and we do our DNA extraction directly from there. And then that's what we use to go through the molecular process to determine if it is Labratories maculans, and then if it is, then we develop the race ID profile for you. When plating the stems, why we don't do things visually is because there's a lot of stuff that can look like blackleg that isn't. When you take it to the culture level, or on the stems, it can look like blackleg that isn't. When you take it to the culture level, you really see what's happening in there. And in this example here, we probably have a Labratories blackleg culture and a Labratories maculans culture, but we won't just rely on the visual difference between that. We'll actually still do the DNA extraction from each, and we can only carry forward the race profiles from the Labratories maculans positive samples. For Verticillium, we can use the same stems that we test the blackleg off of, but instead of having to do a culturing step, we can do a direct DNA extraction right from the stem itself. In the stem, we go slightly above where we take the cutting for blackleg, and we're getting into the vascular tissue. We're taking a cross section of the stem. That tissue gets placed into a tube, it gets ground up into a fine powder, and the DNA is extracted from that. And then we run our Verticillium longiform specific molecular markers against it. So if it's positive, we'll see amplification on our qPCR. We also include a internal control that targets canola DNA, so that allows us to know that if that amplifies, but none of the pathogen-specific markers amplify, we know it's still a valid result because we have amplifiable DNA present. And then if we also include a Verticillium genus marker as well, so it amplifies all genus of Verticillium. So for us to call a sample positive for Verticillium longiform, that has to amplify as well as the Verticillium longiform specific molecular marker. We then note down the number of stems that were positive, write down the reports, and those results go out at the same time as the blackleg results. So for the blackleg race ID test, it was actually developed at Agriculture and Agri-Food Canada here in Saskatoon. So it is their protocol that we follow. We had to do a validation before we were allowed to start testing for the public, and so we've completed that. We've completed the Canola Council of Canada's ring test for verticillium detection. So they sent us a bunch of blind samples that had different diseases in there, and we had to tell them what was in there. So we've had the molecular markers that we use for the test validated by external sources. So we're very confident in the results that come from our testing. And we've also completed the ring test for clubroot detection in soil. And that one was actually down to the quantification level. So when we do find clubroot in the soil, we can report back to you the number of spores per gram that are there, because that's really important for your management practice on your farm moving forward. It's important to note that you don't have to wait till late season to survey your field for verticillium. Even though you may not see the visual symptoms, we will find it in the lab. This was a sample that was submitted earlier on in the program, so the stems are still fairly green. Some of them do have some of the symptomology of verticillium, being the kind of bleaching, damping off of the one side of the stem. But when we take our cross section of the vascular tissue from here, we can, if there is verticillium present, we will find it and we will see it on our PCR. And these samples, these stems were all positive for verticillium, even though they don't have the classic visual symptoms. So when you're going out to scout for blackleg, you can still get the verticillium results from it. You won't just necessarily see it. It's a little easier to scout when the stems are green for blackleg, a little easier in the field. When they kind of get older, it gets harder to visually tell what's going on, because a whole bunch of other things can move into the stems that aren't necessarily pathogenic at all, but they change the appearance of them and make you think that you have maybe more of a problem than you actually do. For a sample like this one, this was submitted under the same program, but these samples would have been taken later on in the season. As you can see, they're starting to gray off. These were probably submitted thinking that this would be verticillium, but just because it's gray doesn't mean it's a verticillium. There's a lot of things that can actually cause that. Saphrites after harvest or other diseases. In this case, this is actually blackleg. The difference on the stem would be the pycnidia and not the microsclerotia that are the kind of visual diagnostic for verticillium. But you don't have to wait for your stems to turn gray or go scout this long. We will find the verticillium in there even if the sample's green. And just because you're seeing graying does not mean it's verticillium. This sample had no verticillium in it. It was all blackleg. And then we have samples that were taken later, but this is a good visual representation of what happens when it is verticillium. You really start to see the epidermis start to shred, and underneath that you will see the microsclerotia. They're very small, they're smooth, and they'll run up and down the vascular tissue of the plant. And then through the testing program, we also have samples that will have both. So this sample had both blackleg and verticillium on it, and we were able to find it even though there's not really the visual indicators right now of verticillium, just based on where we take our sections. But don't be surprised if your results come back with the presence of both blackleg and verticillium on the samples. So when we receive a soil sample through the SaskOilseeds Clubroot program, we take a portion of the sample, it's air dried, it's sieved, so it's all a uniform size. Then we take two approximately 250 milligram replicates and do a DNA extraction directly from the soil. Once we have the DNA, we run our Clubroot specific molecular markers against it and include an internal control for a soil bacteria that's kind of present in all soil samples. That's what we use to make sure that we're not reporting any false negatives. If the Clubroot specific molecular marker amplifies, we can then compare that to a standard curve that we've received from researchers in Alberta that gives us our known quantities of clubroot in soil, and that's how we can provide you with the number of spores per gram that are in your field. So you won't just get a detected, not detected result. If we do detect it, you will get the number of spores per gram.
Once you get your disease testing results, for blackleg, it'll give you a list of a couple races you might see in your field or some specifics, and if you have blackleg. If you don't have blackleg, probably that means that whatever blackleg management you're already doing on your farm is working. We have lots of different tools to reduce the blackleg presence, but resistance is one of the biggest tools that's really effective in driving down blackleg numbers and limiting the yield loss. Verticillium is just a presence stand, so whether you have verticillium or not, we do know that there's a link between verticillium and blackleg. Quite often if you have verticillium and blackleg, yield loss seems to be worse. So while we don't have a lot of tools in verticillium, if you do have verticillium in your fields, doing an extra good job to control your blackleg or keep on top of your blackleg management may limit some of the yield loss from verticillium. A lot of research is ongoing to hopefully create best management practices that we can use in the future. SaskOilseeds will reach out to the growers with the results of the tests from the seed lab. In addition to that, we also sent blinded results to Ag Canada and to research places so they can use information. So the blackleg results are going to go to Ag Canada. They use that to do some monitoring and in their ongoing research of blackleg. For verticillium, that'll go to the ministry. Again, it's blinded to the RM level. Nobody knows where the samples come from except for the RM level, but that goes towards the ministry's map that they make. So every winter you might be familiar with a new verticillium map, showing where verticillium is in the province, if it's spreading, where it's maybe getting worse. And that comes with partially from results from the disease testing program. The SaskOilseeds disease testing program is available every fall from late July, August, all the way through October. For producers that want more information on how to apply, go to the SaskOilseeds website. It also gives information on how to take the samples and where to send them.